Welcome to this video wherein we are going to talk about enzymes and its classification. Enzymes, we come across this a number of times, whether you are dealing with diseases, whether you are dealing with drugs, whether you are dealing with normal metabolism, enzymes are there everywhere and you need to have a clear idea of what they do, how they work, how they behave, who irritates them, who inhibits them, who kills them, or what does it kill. Okay. So, enzymes, these are catalysts. Enzymes are catalysts. They increase the reaction rate. They increase the reaction rate by decreasing activation energy, and doesn't change the Gibbs free energy of the reaction. The Gibbs free energy of reaction, or delta G0. Complicated word, but it means that when a substrate is converted to a product, certain amount of energy is released. Okay, that energy is not changed by enzymes. What does it change? It changes the activity or activation energy. That means that every reaction requires some energy to take place. Okay, and if you add an enzyme, that reaction energy will come down. Okay, or the activation energy for that reaction will come down. I will explain this with a simple graph here. Now, this part is your energy. This part is your energy, all right. And imagine that this is the basic energy levels at which the substrate is present. Now, if you add the enzyme, a lot of energy is required for it to get activated, to form the product. Once the product forms, this is the product that is formed. And this is your delta G0, that is your Gibbs free energy, or the energy that has been produced during the reaction, that is conversion of substrate to product. This energy here is your activation energy. It is your activation energy. So, this much energy is required for the substrate and enzyme to get for the substrate and reactants, or for the reactants to do the reaction and form the product. So, this much energy, unless this much energy is there, the reaction will not occur. Now, when you add an enzyme, what is happening is the activation energy that is required, earlier say 100 kilojoules of energy was required, but now when you add an enzyme, what happens is the activation energy will come down. Now, this will be your activation energy. So, what are enzymes doing? They are decreasing the activation energy. They are decreasing from here to here. They are decreasing the activation energy, but they are not changing the energy that is released, okay, during the reaction. That is, the free energy change that occurs during a reaction will not change by an enzyme. Okay. So, this one is not changed whether with enzyme or without enzyme, but this without enzyme, this is with enzyme. Okay. So, that is the thing that enzymes do. They are catalysts, they increase the reaction rate, decrease activation energy, and there is no change in the Gibbs free energy of reaction or delta G0. That is what it means. Okay. Decreases activation energy from here to here, but no change in the energy, total energy of the reaction or the energy liberated from the reaction. Now, let us see how enzymes are classified. Enzymes classification. So, enzyme classification, enzymes are classified broadly into 7 classes. Earlier it was 6, now 7 classes. 2019 a new class of enzyme has been added. Now, these 7 classes have been classified, and International Union of Biochemists, they have given this classification. That classification is also called as EC classification. It is also called as EC classification. This EC classification is again, or it divides the classification into class, subclass, subclass, and a serial identifier or a serial identifier. Class, subclass, subclass, and serial identifier. Now, this EC classification, every enzyme will have a number EC1 divided period, another period, another period. So, this is one number, second number, third number, fourth number. The first number represents the class, the second number represents the subclass, the third number represents the subclass, and the fourth number is the serial identifier. Now, what is this class? Class denotes the type of reaction. Class denotes the type of reaction. What type of reaction is this enzyme doing? Okay. So, either it is oxidoreductases or transferases, liases, like that. Subclasses defines the type of group, involved, the type of group or reaction involved. That is, it can be either oxidases, reductases, hydrogenases. So, it can be either an oxidation, reduction, hydrogenation, like that. Subclass defines the type of bonds generally, okay, monooxygenase, dioxygenase, like that, types of bonds. And serial identifier will define the cofactors and substituents that are there, cofactors and substituents that are there. Okay. So, a class, subclass, subclass, and a serial identifier. So, EC, there are 4 numbers after EC, 1, 2, 3, 4. This denotes class, subclass, subclass, and your serial identifier. Okay. Now, let us look at the various classes that are there. I have prepared a mnemonic, or it is not my mnemonic, I have taken it from, I was listening to a class, and that person, God bless him, he had given this. I have slightly modified it. Okay. On the Himalayas, Life is lightning thunder. Life is lightning thunder. Okay, this is the mnemonic. Okay, it's about the adrenaline rush when you go try to climb the Himalayas. On the Himalayas, life is lightning thunder. So class 1, class 2, class 3, 4, 5, 6 and 7. Okay. Class 1 on O, oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and finally translocases. Now let us look at what do they do. Oxidoreductases transfers electrons from one group to another. So one group to another. Examples for these are your hydrogenases, lactate hydrogenase, succinate hydrogenase, alcohol dehydrogenase. Okay, all those hydrogenases and dehydrogenases will come in this. Then oxidases and reductases, monoamine oxidase like aldehyde reductase, monoamine oxidase, all those are examples for that. You didn't write all those examples, just write hydrogenases, oxidases, reductases. Okay, so those compounds form oxidoreductases. Next, what are transferases? Transferases are the ones which transfer a functional group from one product to or from one substrate to another. Okay, transfer a group. That group is mostly a phosphate, methyl, and an amino group. A phosphate group, methyl group and an amino group. They just transfer from here to there. Okay. So what are they? Those which transfer a phosphate groups, we have seen many kinases, phosphatases. What they do is they either remove or add a phosphate group. Okay, kinases add a phosphate group, phosphatases remove a phosphate group. We will see that in detail when we are talking about enzyme regulation, especially in glycolysis, wherein these enzymes are either phosphorylated or dephosphorylated. Phosphorylation is done by kinases, dephosphorylation is done by phosphatases. Kinases are activated by glucagon, phosphatases are activated by insulin. Okay, so that is how glucagon and insulin they balance the blood glucose regulation. We will talk about that in detail. I will make you by heart that. Okay, for now you remember that transferases are the ones which transfer a group like phosphates, methyl and amino group. So the examples for these are kinases, phosphatases, Next, transmethylases. Transmethylases always work with S-adenosyl methionine. Remember, active methionine and active sulfate. Active methionine is S-adenosyl methionine. It is the methyl donor of the body. PAPS is the sulfate donor. Okay. Okay. Will not deviate from here. Kinases, phosphatases, transmethylases, which always works with S-adenosyl methionine, and transaminases and transaminases. Okay. Next, hydrolases. Hydrolases are those which cleave a bond. Hydrolases are the ones which cleave a bond in presence of water or in a water medium. So they require, hydrolysis are the ones which require a water medium. Water should be there for it to act properly. What are those? Glycosidases, peptidases, nucleases. Basically all your digestive enzymes will be there in this class, hydrolases. Next is lyases. Lyases, these break a bond. Lyases, they break a bond by adding or removing a functional group. Like water, carbon dioxide, ammonia. Here there is a group transfer, no hydrolytic cleavage of any bond. Here the bonds are being broken by either addition of a group or removal of a group. What can that group be? Water, carbon dioxide and ammonia. So examples for these lyases are hydratases, dehydratases, decarboxylases, aminases and deaminases. Okay. So what are lyases? They break a bond by adding or removing a group of water, carbon dioxide or ammonia. Isomerases. They bring about structural change. Structural changes forming isomers. Structural changes forming isomers. Examples are triose phosphate isomerase, like that. Isomerases, epimerases and racemases. Isomerases, epimerases and racemases. Next, ligase. Ligases, they join a bond, they join substrates to form a bond using energy, like your ATP, GTP, like P or GTP. Examples are synthetases, Then your carboxylases, all those are examples for your ligases. Translocases transfer, they catalyze transfer of substrates like hydrogen ions, transfer of groups like hydrogen ions or anions, inorganic anions, cations, amino acids, then carbohydrates and their derivatives. Transfer from one place to another, not from one substrate to other substrate. Transfer from one place to another. Here transfer from one substrate to another substrate. Here translocases from one place to another place, from inside the mitochondria to inner membrane space, or from the matrix of mitochondria to the inner membrane space, from the cytoplasm to the mitochondria, from the endoplasmic reticulum to the cytoplasm, like that. So translocases, what they do is they transfer catalysis or they catalyze transfer of protons or inorganic anions and cations, amino acids, carbohydrates from one place to another. Examples are ATP type sodium transporter, ATP type sodium transporter, your voltage gated channels, all these are examples of translocases. Okay. So that is about the classification. I have made it as simple as possible. I bet nowhere it is given like this. So simplified totally, on the Himalayas life is lightning thunder. Oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases and translocases. Now what does oxidoreductase do? Transfer one electron from one group to another. Okay, they just shuttle the electrons. Examples are hydrogenases, oxidases, reductases. Transferases transfer a group like phosphate, methyl and amino group. Examples are your kinases, phosphatases, transmethylases and transaminases. Hydrolases cleave a bond in a water medium. A water medium has to be there for it to cleave. Examples are your glycosidases, peptidases, nucleases, all your digestive enzymes come in this category. Next, lyases, they break a bond by adding or removing a group, that is it can be either a water, carbon dioxide, ammonia. Examples are hydratases, dehydratases, decarboxylases, aminases, deaminases. Now you may have a doubt, hydrolases cleave a bond in a water medium. Okay. Here hydratases and dehydrotases, they are lyases, not hydrolases, but both the places you will find water. Here the water is water medium, like swimming. Okay. The person, suppose you are an enzyme, you are swimming in water. This is hydrolase. Here you are either drinking or passing urine. Hydratases, dehydratases. Understood the difference? Hydrolases swimming in water, everywhere water. Enzyme is swimming in water. Here lyases drinking water or taking out water. So hydratases and dehydratases, that is the difference between them. Okay. Lyases break a bond and all that. Isomerases, they bring about structural changes. Ligases and translocases. Okay, with this we finish the classification of enzymes. If you write this much, more than enough. The examiner is really happy, and I bet you will score maximum with this. Okay, you needn't do anything beyond this. So this question, classification of enzyme, is very important for your board exams and also for your PG entrance exams. Okay, so you can make a chat of this, keep it somewhere in your books or on the top of your book or on the top of your bench or on the ceiling where you are sleeping. You can do things like that, crazy things like that, like I used to do. All right, anyways, so that is about the classification.